Hi friends, welcome to Study SSC. Today we are going to start a new series which will be helpful for the upcoming Ssc Cgl, Chsl Mts and other related exams of 2024. This series is provided in a concise manner, so you will find it easier to take down the notes and to learn it. And the first topic in our series is science. And in science, we are going to learn biology, and chapter 1 is cell. Now, let's jump into the video. Cell. The three definitions of cell are: cell is known as the building block of life. It is also known as fundamental unit of life or the structural or functional basis of life. Now, let's learn about the discovery of cell. The study of cell is known as study of cell. Study of cell is known as cytology. Study of cell is known as cytology. And who is known as the father of cytology? Father of cytology is Robert Hooke. And the first one to discover dead cell in core was Robert Hooke in 1665. He discovered dead cell in core. And he is also known as the father of cytology. And cytology is the study of cells. Next is living cell was found in pond water in 1674 by Anton von Leeuwenhoek. Living cell was first discovered by Anton von Leeuwenhoek in pond water in 1674. Who discovered nucleus? Nucleus was discovered in 1831 by Robert Brown. Nucleus was discovered by Robert Brown in 1831. The molecule discovery of DNA was first done by Friedrich Mischer in 1869. Friedrich Mischer discovered the molecular discovery of DNA in which year? 1869. And we all have heard about the double helical structure of DNA. This is very important. Double helical structure of DNA was discovered by Watson and Crick. Double helical structure of DNA was discovered by Watson and Crick in 1953. Camillo Golgi discovered Golgi bodies. Golgi bodies was discovered by Camillo Golgi. Golgi bodies was discovered by Camillo Golgi. Friedrich Mischer discovered the molecular discovery of DNA in 1869. Double helical model of DNA was discovered by two scientists, Watson and Crick in 1953, and Golgi bodies were discovered by Camillo Golgi. There are two types of cells. They are the prokaryotic cell and the eukaryotic cell. Prokaryotic cells are unicellular, small and simple, whereas eukaryotic cells are large, complex and multicellular. There is no nucleus in a prokaryotic cell, and in a eukaryotic cell there is nucleus. That is, nucleus is present in the eukaryotic cell and nucleus is not present in a prokaryotic cell. And in a prokaryotic cell we can see circular DNA, and in eukaryotic cell we can see linear DNA. Prokaryotic cell does not contain a membrane and eukaryotic cell contains membrane. Membrane is present in eukaryotic cell. The examples for prokaryotic cell is bacteria, mycoplasma, cyanobacteria. Mycoplasma is very important. It is the smallest known bacteria. The smallest known bacteria is Mycoplasma. The examples for eukaryotic cell are plant and animal. All plant and animal cells are eukaryotic cells. Parts of cell. The first part of a cell is the cell membrane. The cell membrane provides a definite shape and holds content. It is flexible, thin and elastic. Cell membrane is composed of proteins and lipids. This is important. Cell membrane is composed of proteins and lipids and they are selectively permeable. Next part of the cell is the cell wall. The cell wall provides shape, strength and turgidity to the cell. It is freely permeable and it is also non-living. In plants, the cell wall is made up of cellulose. In plants, cell wall is made up of cellulose. This is very important. In bacteria, the cell wall is made up of a substance called peptidoglycan. In bacteria, cell wall is made up of peptidoglycan. And in fungi, the cell wall is made up of chitin. Chitin. In fungi, cell wall is made up of chitin. Next part of the cell is the nucleus. Nucleus is called as the brain of the cell. The components of nucleus are nuclear envelope and nucleoplasm. The nuclear envelope is composed of membrane and it covers the nucleus. The first part is nuclear envelope and it is composed of membranes and it acts as a covering of the nucleus and this separates nucleus from cytoplasm. Okay. It separates nucleus from cytoplasm. Next component of the nucleus is the nucleoplasm. Nucleoplasm is the liquid ground substance of nuclear envelope. The liquid ground substance of nuclear envelope is called as the nucleoplasm. The two types of nucleoplasm are nucleolus and chromatin material. Two types of nucleoplasm are nucleolus and chromatin material. This nucleolus can be one or more in number and it forms protein forming centers called ribosomes. This is very important. Protein forming centers are ribosomes. The second type is the chromatin material. Chromatin material consists of entangled mass of thread-like structures. Entangled mass of thread-like structures and chromosomes. These chromosomes are thread-like structures that is visible during the cell division. Thread-like structures visible during the cell division is the chromosomes and the components of chromosomes are, there are two components of chromosomes. They are DNA and proteins. First component is the DNA. DNA consists of the genes and the second component is protein and we have seen that protein synthesis takes place in ribosomes. Okay, there are 23 pairs of chromosomes and in these 23 pairs, 22 pairs are autosomal and one pair is the sex chromosome, that is XX and XY chromosome. Next part of the cell is the cytoplasm. Cytoplasm is the part between the plasma membrane and the nuclear envelope and cell organelles are present. The part between plasma membrane and nuclear envelope is known as cytoplasm. The aqueous ground substance present in the cytoplasm is known as cytose. CYTOS. Aqueous ground substance present in the cytoplasm is called as the cytose. The next topic is cell organelles. First cell organelle is the mitochondria. Mitochondria is known as the powerhouse of the cell. This is very important. Which part of the cell organelle is known as the powerhouse of the cell? Answer is mitochondria. The adenosine triphosphate synthesis takes place in mitochondria. That is ATP synthesis. Full form of ATP is adenosine triphosphate. ATP is known as the energy currency of the cell. Next organelle is the lysosome. Lysosomes are known as the suicidal bag of the cell. This is also important. The suicidal bag of the cell is lysosomes. I forgot to mention one point in mitochondria. Mitochondria possess its own DNA and ribosomes. Mitochondria possess its own DNA and ribosomes. The next organelle is the Golgi bodies. The function of the Golgi bodies is to store and modification of package. Golgi bodies forms the lysosomes, and there are two phases in Golgi bodies. The cis phase is towards the nucleus and the trans phase is towards the plasma membrane. The next cell organelle is the endoplasmic reticulum. Endoplasmic reticulum is of two types. That is, first one is the rough endoplasmic reticulum and second one is the smooth endoplasmic reticulum. The rough endoplasmic reticulum makes proteins. That is, it helps in the synthesis of proteins, and ribosomes are present in the rough endoplasmic reticulum. As seen earlier, ribosomes are associated with protein synthesis. The next is the smooth endoplasmic reticulum, and it helps in the synthesis of fats and lipids. Ribosomes are absent in smooth endoplasmic reticulum. Next organelle is the vacuoles. Vacuoles are present in plants and in animals. In plants, it is large, distinct and permanent, whereas in animals, it is small and temporary. And in plants, it provides turgidity and rigidity to the structure. In animals, vacuoles helps to store glucose, glycogen and protein. Next organelle is the plastids. Plastid is found only in plant cell and it is absent in animal cells. It has its own DNA and ribosomes. In mitochondria also, mitochondria possess its own DNA and ribosomes. The three types of plastids are chloroplast. Chloroplast stores the chlorophyll pigment. The second type of plastid is the chromoplast. Chromo means color. It helps to store the color pigment. And the third one is the leucoplast. Leucoplast helps to store starch, oil and protein granules. These three are important. The three types of plastids are chloroplast, chromoplast and leucoplast. Next topic is the cell division. The two types of cell division are mitosis and meiosis. First one is mitosis. Mitosis occurs in somatic cells, and in mitosis, nucleus divides once. The division is diploid and two daughter cells are formed. While in meiosis, it occurs in germ cells and the nucleus divides twice, and meiosis is haploid and four daughter cells are formed. The phase of cell division are the G1 phase. G1 phase is associated with the growth. The G2 phase. G2 phase is associated with the growth and preparation for mitosis. The S phase, which is associated with the DNA synthesis, and the M phase, which is associated with the mitosis. Thanks for watching and hope you all have understood the class. Thank you.